something that is known as the end replication problem, the inability of the DNA machinery in the cell to replicate the chromosome right until the end. So every time the cell is dividing, the DNA is replicated before the cell is dividing to two daughter cells, we are losing these small segments at the end of the DNA, okay, which are the chromosomes. And eventually, with increased cell division, the telomeres become critically too short, and that signals the cell to stop divide, and eventually lead to the DNA damage response and to one of the hallmarks of aging, which is cellular senescence. Okay, this is replicative arrest. And this is a normal process, okay, and this is how our cells getting older as we get older. On average, if you will measure the telomeres in a 20-year-old versus a 40-year-old versus a 60-year-old, you will see, on average, shorter telomeres. And this is because our cells divide in many more times. Okay, so this is something why telomeres are considered to be this kind of a clock that regulates cellular division. But in recent years, there is evidence that telomeres are malleable and are responsive to the environment, and specifically that stress can accelerate the erosion of telomeres. So I want to give you just a few examples. This is just one example in relation to telomeres and early mortality. This is the first study published in 2003 by Richard Cawthon and colleagues. There are more recent studies, bigger studies showing the same effect. But in this initial study, they had individuals aged 60 to 74 and 75 or older. They measured the telomeres, they did a median split, and they showed that those individuals with shorter telomeres on average had poor survival rate. Okay, they had a threefold higher risk to die from cardiovascular disease and eightfold higher risk to die from infectious disease. Okay, so that was the first example. The telomere length can even predict early mortality. Okay, and there are more recent studies that are showing this same effect. And in relation to stress, this whole field really started with this seminal study by Lisa Eppel and colleagues, showing that telomeres actually can be responsive to psychological stress. Okay, in this study, the participants were women who were mothers of a chronically ill child. And what they showed, and this is the main finding, is that the chronicity of caregiving, the longer they had to take care of the child, was associated with shorter telomeres. Okay, so this is was the first example that chronic psychological stress is related with shorter telomeres. Okay, and this is a conceptual model that we developed that kind of summarized the lifespan influences on telomere length. We have the prenatal adversity and childhood trauma that can lead to faster telomere erosion. There is unidirectional errors, meaning that this goes only in one direction. Later in life, we have adult mental disorders and age-related diseases that can also impact the rate of telomere erosion, and that actually can go in both ways. We don't really know the causal direction. It could be that short telomeres are causing the onset of diseases, or it could be that the health status itself is affecting the rate of telomere erosion. Probably go both ways. And there's also indication, what you see in the top, that healthy lifestyle factors can mitigate some of the adverse effect on telomeres. And there's a lot of research. Really, this is a field that's really been exploded in the last 10 years with a lot of studies showing association with many health risk factors and depression and PTSD and anxiety disorders, showing this kind of link with shorter telomeres. But here I'm going to focus just on the first two boxes, on the prenatal adversity and childhood trauma. Okay, so we'll start with the early life stress effect on telomere erosion. And briefly there, I'm going to show you a few previous studies. This is the first study to document this effect by Audrey Tirka and colleagues, showing that adult participants who reported emotional and physical neglect had shorter telomeres. That was the first preliminary evidence for this effect. And then many studies followed, showing the same kind of effects, that childhood trauma is associated with shorter telomeres. The last study at the bottom by Stacy Drury and colleagues was the first study in children to document this effect that poor institutional care was associated with shorter telomeres in young kids. Now, even though this is just a sample of the studies, there are more studies showing the effect of early life stress on telomeres. They still had several limitations. And the main ones is that almost all studies looked at adult participants who retrospectively recalled incidents of trauma in childhood, and we know that there is bias in these types of assessments. All studies use cross-sectional designs. Okay, so they were not able to look at telomere change over time, which is a really important marker, rather to look just at length at one time point. And importantly, the last point, it was not known whether telomeres began eroding during stress exposure in childhood, or whether the erosion occurred years later as a consequence of the health problems that it might have developed. Okay, so we know from the previous talks that early life stress is associated with many health problems, mental health problems, and these health problems are associated with shorter telomeres. So maybe these are the processes that are related to faster telomere erosion and not stress per se in childhood. So we try to overcome some of these limitations by using prospective longitudinal design. And this is from the environmental risk or e-risk study, which is a representative birth cohort in the UK. More than 1,000 twin pairs, half of them monozygotic, half of them dizygotic. And you see at the bottom the design of the e-risk study with the data collection ages circled in blue. So we started at birth, and then age 2, and then 5, 7, 10, 12, and the recent phase was just ended recently at the age 18. And I'm going to show you data from age 5 and 10. Okay, so we collected DNA at both of these ages from buccal cells. And we had assessments of early life stress between ages 5 and 10. Okay, we assessed 3 types of violence exposure. Either domestic violence between the mother and a partner, frequent bullying victimization, and actual physical maltreatment by an adult. So first I'm going to show you each type of violence by itself in relation to telomere erosion between 5 to 10. And this is actually really relevant to what Beck mentioned about the specificity of specific stressors. So this is the effect with, before that I just wanted to kind of show you some of the types of violence exposure that we had in our study, and we are talking about really severe types of violence exposure, the domestic violence, the bullying, the physical maltreatment, really severe cases. Okay, so the results with the domestic violence. The kids were exposed to domestic violence in red versus the control who were not exposed, and as you can see, there's slightly faster erosion of telomeres from age 5 to 10, okay? But that was not significant. When we looked at bullying victimization, actually it doesn't seem like the telomeres are eroding faster. We had shorter telomeres at baseline at age 5 and also shorter at age 10. And the same was true with physical maltreatment. Here we do see slightly faster erosion, and again, shorter telomeres at baseline and at follow-up, but none of these violence exposure by themselves were statistically significant, okay? So then we combined all of the violence exposure to form this kind of a cumulative index, and this is really based on the evidence that the effect of stress may be seen more clearly when measured in a cumulative way. And this is especially true when we are talking about telomeres. Really, most of the effect on telomeres is when you have this kind of cumulative index and you have assessments with chronic stress over time. So this is the effect of the cumulative violence exposure, and this is a study that was published a couple of years ago. And here what you see is that only the kids who were who experience 2 or more types of violence exposure. And maybe this is also emphasizing kind of dysfunctional families, but only these kids had faster erosion of telomeres between 5 to 10 compared to the kids who were exposed to just one type of violence or kids who were not exposed to violence at all. Okay? One of the limitations of the study is that what you see at baseline at age 5 is that kids who were exposed to any type of violence already had shorter telomeres at baseline. Okay? And we suspected that some of these violence exposure had began before age 5. Okay? And this is probably why they already have shorter telomeres at baseline, but still they had faster telomere erosion. So this is important because that was the first study to document the effect of telomere change over time in childhood while children are actually experiencing distress and before they develop all of the physical health outcomes. Okay? So at the age 10, the kids were still healthy, but they still had faster telomere erosion, so that might be as a mechanism that can explain the later health outcomes. Okay, so that was the first study in relation to early life stress. The second one that I'm going to show you is in relation to perinatal adversity. Okay, and this is from a different prospective study, and it's related to the developmental origin of health and disease model. So this was the first study to show that stress exposure in utero can be linked with shorter telomeres. That was by Sonia Entriger and colleagues. They had adult participants and they interviewed the mothers about incidents of trauma during pregnancy, and they assessed really severe types of stressors, and they measured the telomeres in the adult participants. And what they showed is that those participants who were exposed to stress in utero had shorter telomeres later life compared to the control group. Okay, so that was the first example that stress in early life can already have this kind of impact on telomeres. So our study was slightly different. We looked at common perinatal complications, and we used a prospective study to test this hypothesis. So in order to show you the results, I need to take you to the other side of the world, to a small city in the South Island of New Zealand called Dunedin. If you missed Andrea Denise's talk yesterday, I'm just going to repeat briefly what the Dunedin study is. So we are talking about a birth cohort of more than 1,000 individuals, all born in 1 year, between 1972 and 1973, in the same hospital in Dunedin, in New Zealand. And they've been followed prospectively from birth until age 38. That was the last assessment. This study is still going on. The next phase is scheduled to age 44. And what really is amazing about this study is the last column that you see, the retention rate. So the last phase at age 38, 95% of the living members took part. Okay? And this is really an amazing data about this study. 30 study members have died over the years, but most of them are still taking part. And if you're curious to see how a 4-decade prospective study looks like, so here are 2 study members at birth. They are twins. There are only 4 twin pairs in the study. Here they are at age 3, 5. It's also interesting to see how fashion is changing over the years. 7, 9, 11, it's a classic 80s fashion. 13, 15, 18, 21, 26, 32, and the last phase at age 38. So it's really amazing to see how people change over time. And the amount of information that we have on the Dunedin study members is really amazing. What do we know about them? We basically know about them everything, from personality, psychopathology, physical health, dental health, sexual behavior. And we also have a lot of biomarkers and DNA that was collected over the years. So what I'm going to show you is data that was collected at the first stage, at birth. And we assessed perinatal complication at birth. We included 18 common obstetric problems, both in the mother and the baby, including maternal diabetes, hypertension, low birth weight, very common problems at birth. We had close to 64% who did not experience any perinatal complication, 25% who had one type of perinatal complication, and 88 that had two or more. Okay, we again had this kind of accumulative index of perinatal complications. Our outcomes were at age 38. We had two aging indicators. One of them was telomere length, and the other one was perceived age, which I'm going to explain later. And thinking that this is the Dunedin study, we have all of the information, prospective information from birth to age 38. And we control for all of these factors that can explain the link between perinatal complications and aging indicators, which include family history of medical problems, childhood social adversity from birth to age 11, cognitive health, so this is IQ, average from 7 to 38, mental health, again, this is a cumulative factor known as the P factor from age 18 to 38, vascular health, both systolic and diastolic blood pressure, and physical health. This is another kind of accumulative index that we have in Dunedin. So these are all of the factors that we control for. And when we looked with the association between telomeres and between perinatal complications and telomeres, that was the finding. Okay, we saw this kind of a dose response effect. Increasing number of perinatal complications was associated with shorter telomeres at age 38. Okay, and that effect was still held when we control for all of these factors that I showed you. So that was an example that related to the developmental origin of health and disease, that really adverse exposure in the earlier stages of life can be associated with aging indicators in midlife. The second aging indicator that we use is kind of peculiar. This is a perceived age measure. And this is an indicator that clinicians are actually using. And these are two studies conducted in Denmark. They used a very powerful design of identical twins. They had assessors that rate the perceived age of the twins, and they showed that the older looking sibling had poor cognitive function, and it also was correlated with shorter telomeres. Okay? And in another study, they actually followed them over time, and they showed that the twin who looked older also died earlier. Okay? So perceived age can actually be an indicator, general indicator of health status. So in our study, we had two measures. Already showed you the results, but in our study, we had two measures of perceived age. We had independent panel of raters that were blind to the actual age of the study members, and they had to assign an age range from 20 to 24, 25 to 30, up to 65 to 70. Okay, so that was the age range factor. The second one was assessed by an independent panel of assessors, and they all knew that all of the study members are 38 years old, and they had to assign a relative age from 1 young-looking to 7 old-looking. Okay, and we then standardized both scales, and we averaged them to create the perceived age scale in the Dunedin study. And these were the results. Again, the same pattern as you saw with the telomeres. More perinatal complications in a dose response manner was associated with older perceived age. Okay? And we also correlated previous studies, and in our study also telomeres were correlated with perceived age. So the shorter telomeres, the older the study members looked like. Okay? So we basically had two aging indicators, both inside and outside, linking stress really early in life to aging and midlife. Okay, so let me just tell you briefly about some of the next steps in our research. So first of all, going back to the environmental risk study. So this is an ongoing study, and I mentioned that the last phase was completed recently at age 18. The study that I showed you was actually a pilot study. It was only 10% of the sample. And we now collected DNA again at age 18. And we have now both buccal cells that we can compare to the earliest DNA collections at 5 and 10, and also blood. And we are now looking at the telomeres, both at ages 5 and 10 and 18. And considering that this is a twin design, both monozygotic and dizygotic, we have discordant cases of violence exposure. So this is a very powerful study to look at the effect on telomere erosion. So this is one of the next steps that we're going to take. Considering the mechanism, so this is really one of the main focuses of my research in the lab. If we think about the effect of early life stress on later risks for age-related diseases, it seems that telomeres may function in this kind of master integrator, linking the adverse effect in early life with later health outcomes. It's still not clear whether the telomere is an actual causal mechanism, but it can be sort of like an indicator. But importantly, we really don't understand the mechanism, how stress is causing telomeres to erode faster, and how short telomeres are causing disease, if they are causing disease. Some of the mechanisms can be biochemical stressors in the cell and epigenetic regulation. There's very active research now trying to understand some of the biological mechanisms that regulate telomeres. Some of the systems that I'm focusing in my lab are all of these biological embedding factors. If we're talking about stress, so the HPA axis with end product cortisol, oxidative stress, inflammation, telomerase, which is an enzyme that can maintain and protect the length of telomeres. All of these processes, factors likely work on different pathways. So for example, oxidative stress and the relation between oxidative stress and antioxidant defense can work directly on the telomeres. Inflammation or inflammatory factors can cause cells and stem cells to divide. And as a consequence, we will have shorter telomeres because of the end replication problem over time. High levels of cortisol, chronic level of cortisol can downregulate telomerase. And there's also a receptor for cortisol in the mitochondria. And the mitochondria itself seems to be a very important factor that regulates telomeres. So there's many different factors that likely operate in different ways that can regulate telomeres. And there's really a lack of understanding currently about which factor is more important. And it's probably all of these factors together. And before I finish, I just want to end with this notion, which is kind of important, that this process is not inevitable. Okay, there's already been discussed that maternal sensitivity is a really important factor that can regulate telomeres, and these are very recent studies showing that parental care can buffer the effect of stress on telomeres. So telomere length can be a good biomarker to test intervention efficacy through sensitive parenting. Okay, so there's still hope. And I just want to end with just acknowledging my colleagues and to thank you. Thanks.
Any questions? We can take a question now before our coffee break.
Hi, Idan. Great talk. Thanks. Quick question, kind of coming back to our adaptation theme. With our shortening of telomeres, is there any association with earlier onset of puberty, for example, or some other adaptive advancement?
Good question. No, we don't know that, but we actually have a grant that we just submitted with Jay Belsky testing that specific hypothesis.